guys, welcome to my YouTube channel. I'm Dr. Shashita Verma and today I'm going to discuss about another topic, that is restriction endonucleases and its nomenclature. So basically we're going to talk about the nomenclature, the naming system of these restriction endonucleases. So let's get started. Restriction endonucleases. Now these endonucleases, they are named based on the organisms in which they were discovered. Like for example, if I have discovered one restriction endonuclease from E. coli, then the name will be based upon this organism, that is E. coli. So this is how scientists have done. Let me explain to you with the help of an example. Now we have a restriction endonuclease, that is ECO R1. We have this restriction endonuclease, that is ECO R1. So what does this ECO R1 stands for? This ECO R1 is written in italics. It is being italicized. Why? Because it has the genus and the species name. Now ECO R1, it is pronounced as ECO R1. Alright, it is pronounced as ECO R1. This is the pronunciation of this restriction endonuclease. So to start with, we'll first see what does this E stands for. If we see clearly, then it is visible that this ECO is an acronym and a three-letter. Now E stands for Escherichia, that is the genus of the organism. CO, that is co. Co stands for coli, and this is a species. So this three-letter, this is an acronym for the genus and the species of the organism. So it is, we call it as ECO. Now writing about the genus, when we are writing the genus, this E should be capital. This is the first letter of the restriction endonuclease. Then followed by the second and third letter. These are written in small letters and it represents the species of the organism. Coming up to the next letter, R. Now this represents the strain of the organism. The full name of the strain is RY13, but then we will only use the first letter of the strain, R. And 1. 1 stands for the first identified. That means that it represents the order of identification in this particular bacteria, in this particular organism. Yeah. So that together makes ECO R1 and is pronounced as ECO R1. Alright. So let me recapitulate what we have studied. That restriction endonucleases, their names are based on the organisms from which they are being discovered. The first letter that has to be written in capital represents the genus. Like in case of ECO R1, it is E stands for Escherichia. The second and the third letter accounts for the species name, that is CO from coli. R is the type of strain, that is the full name of the strain is RY13, but then we'll take only the first letter of the strain, that is R, and 1. 1 accounts for the first identified, that is in which order the enzyme has been obtained or discovered in the particular organism. Similarly, we have different examples such as BAM H1. Similarly, we have examples here like BAM H1 and HIND3. So in case of BAM H1, this B stands for bacillus, M stands for amyloleucophysis, H is the strain, and 1 is the order of identification. So now B it stands for bacillus, and M it stands for amyloliquefiscence, H is the strain and 1 is the order of identification of the enzyme. Then similarly, if I consider the example of HindD3, then H stands for Haemophilus, In is the name of the species, then stands for influenzae. D again is the strain, and 3 is the order of identification. So you can see here with Bacillus amyloliquefiscens, because this is the genus in the species, so here this 3-letter acronym will be written in italics. Similarly, in Haemophilus influenza, this HIN 3-letter acronym will be written in the italics. Now the B is being derived from the genus Bacillus, and AM is also derived from the species. Similarly, in this, H is being taken from Haemophilus and N is being taken from the influenza. So this is how we name different kinds of restriction endonuclease. I hope this is clear.